So I'm delighted to introduce today's speaker, Dmitri Petrov, who comes to us from just down the road at Stanford. And as much as I am trained to and inclined to hate Stanford, being a Cal alum myself, I have to admit that they have some great biologists, including people like Dmitri. So we appreciate you driving up here. Dmitri works on Drosophila, but for those of you who do field work on wild organisms, understand that Dmitri has very broad interests, and he's published on such things as Hawaiian crickets. He's very broadly interested in the genetic basis of evolutionary change, including genome evolution and how organisms adapt to their environment at a genetic level. And I personally like to follow the Drosophila literature closely because I always feel like Drosophila evolutionary geneticists are sort of 5 to 10 years ahead of the rest of the field. And some of this is just because Drosophila as a model organism has all sorts of power that's not available in other systems. But the rest of the world is catching up. The distinction between model and non-model organisms is diminishing, and so a lot of the things that used to be doable only in a few species can now be done on anything. And so I think it's great for those of us who are working on non-model organisms to be very aware of this field. So Dmitri became well known for his work on transposable elements and using transposable elements to show that there was a mutation rate that was driving a reduction in genome size in Drosophila and some other organisms. And so that was sort of using transposable elements as markers of sort. And he's also used transposable elements to study adaptive evolution. And more recently, he's been very interested in adaptive evolution across climatic gradients. I noticed you have a grant that talks, the title of which is Evolution or Adaptation in 6 Dimensions. And, you know, I feel lucky to study adaptation in 1 dimension, so I'm not sure what that's about, but it sounds very interesting. Anyway, Dmitri, thank you for coming.
Thank you. Delighted to be here. Please interrupt me at any time if you have any questions. I have 2 stories to tell, and I can speed it up if need be, so please don't hesitate. I agree with everything that Michael said. One thing is that Drosophila does live in the wild, so even though it's a model organism, it has a natural history. It's something that I'll talk about today. Is there a pointer? Oh, yes. So recently I became very interested in study of rabbit adaptation, and I think most of us, when we think of evolution, sort of share Charles Darwin's view, is that evolution is a slow and gradual process, and that if we're interested in studying evolution, you need to be looking over vast periods of time. And so it's a quote from Darwin about species evolving through the accumulation of small variations over long periods of time. But at the same time, we know that one of the most pervasive observations about natural world is that you can take pretty much any population of organisms, with some exceptions, apply virtually any selective pressure to it, and it will work. Right? So that's kind of somewhat of a unclear why it should work like this, but that's how it works. So we see it both when we apply completely new selective pressures, and we see very rapid adaptation, probably through de novo mutation, de novo variants. So for example, when people apply pesticides to insects, detectable resistance evolves on the scale of years, right? So usually 5 to 10 years, and you already see a resistance in the insect, and at least in some cases, it's likely to be due to new mutations. We also know that adaptation from standing variation, which is I think what people usually think about rapid adaptation, can happen very, very fast. It takes, admittedly, sort of dedication of British breeders, but still you can take sort of village dogs, apply different selective pressures, and you get all this remarkable variety, say, of dog breeds. The same works in agriculture. So we know that adaptation can work fast. So that's just a primary observation. And I think for evolutionary biologists, it's an interesting system to study for many reasons, but one of them that I find particularly compelling for me is that I'm interested in turning evolution in some way into predictive science. And so here, if adaptation works so fast, that there's a chance of of making predictions about how adaptation will work, and the time scale is such that you can live long enough to see the prediction either come to pass or not. So it creates a good model for the study of evolution. So today I'll tell you about two stories about adaptation in Drosophila. One of them is rapid adaptation to pesticides through de novo mutation, and another one, rapid adaptation on seasonal scales at ancient balanced loci. That's the 6 dimensions grant, and I'll explain what 6 dimensions are. That was not planning to. All right, so the first part. When we think about adaptation by de novo mutation, usually we think in the following way. So there's this time, there's this red line is this time of the onset of some new selective pressure, and then you wait until the right mutation appears. Now, usually the right mutation takes a certain amount of time. Most of the mutations that appear are lost right away, but some of them are lucky enough not to be lost right away, and so then they increase. When they appear on some particular genetic background, and so these different color lines are different haplotypes in the genome. They're distinguished by different patterns of polymorphisms. This mutation then, if it's not lost right away, increases in frequency and, let's say, goes to complete fixation to some intermediate frequency. And when it does it, it drags with it the haplotype on which it arose. So here I'm drawing a cartoon where it's completely unbroken haplotype, but it can be, recombination can make it a little bit degraded. But around the site of the mutation, you expect to see a single haplotype increasing in frequency. So this is a classical picture. It's called a picture of a hard selective sweep. You can imagine it working somewhat differently if the waiting time until the mutation that is not lost right away is short. And in that case, the first mutation will arise on some particular haplotype, but then before it gets to high frequency, the next one will arise, and both of them will go to high frequency, and together they will be present at, let's say, at 100% frequency. The total frequency of both of them is 100%, but because they arose independently, they'll arise on different haplotypes, and even though at this position this mutation is fixed, the two haplotypes went up in frequency. If the rate of mutations to this adaptive alleles is much higher, you can have 3, 4, 5, as many as you want. So that's a signature that's called soft sweep. Pennings and Hermansen realized that it's a reasonable possibility and termed it a soft sweep as opposition to hard sweep. Now, when do we expect to see hard sweeps and when do we expect to see soft sweeps? The key parameter, as it turns out, and I'm not going to go into the math, but the key parameter turns out to be just the rate with which mutations arise in the population, which is again which is inversely proportional to the waiting time until the right mutation. So if you take the population size, how many individuals there are, and you say what's the mutation rate to the adaptive alleles, the per generation, that's how many adaptive mutations arise in the population. And if this number is very small, let's say, usually people think that this number, let's say in Drosophila melanogaster, it's 0.01. It means that it takes, on average, 100 generations before one adaptive mutation will arise. It will most likely get lost. Then the next one, so it takes hundreds of generations, right, before this happens. And if that's the case, when the first lucky one arises, it will get to fixation before the next one happens. This we expect to see if this parameter is on the order of 1 or larger than 1, right? So, as I already mentioned, much of the time people assume that this is the truth, this is where we live in the Drosophila populations, or all populations live in this regime, and therefore what we should be looking for is this kind of a signature. And so pretty much all scans for positive selection assume this model, and they look for the signatures of a single haplotype getting to high frequency. There should be a dip in polymorphism, and there are other signatures that come with it. This is something that people generally do not look for, or have not looked for until recently. So so I was very much in the business of looking for hard sweeps, but got a little perturbed by the fact that we start with a theoretical model, we look for the signatures, but we didn't really have really good positive controls. Just like, what does it actually look like when we know that adaptation really did happen? Can we please look at the signature that's left then? And I said, well, we have some cases where we know adaptation happened, and in particular it happened recently. The signatures should be very clear, and selection was strong. And so together with this undergraduate in the lab, Talia Karasov, she's now finishing up PhD at the University of Chicago, and postdoc in the lab, Philip Messer, we decided to look at the signatures that are left by adaptation to pesticides and one particular gene in Drosophila genome called ACE. So humans put out into the environment pesticides of organophosphate and carbamate families, and these chemicals, they're mimics of acetylcholine. So acetylcholine is used in transmission of nervous impulse through the synapse, and when a single transmission happens, it's released into the cleft, and then this enzyme, acetylcholine esterase, cleans it out so that the next one can happen again. Organophosphates are mimics, and then they bind covalently to this enzyme and inactivate it. So repeatedly, multiple insects have evolved resistance to these pesticides by particular amino acid changes in the enzyme, most of them in the active site. So now that they can discriminate away from this organophosphate, they can still act on acetylcholine, not so well. So in the absence of pesticides, these versions are deleterious, and substantially so like 20–30% deleterious effect. But in the presence, the flies or other insects do not die. And we know where these positions are. So when we started looking at Drosophila melanogaster, we found that there are alleles that have particular resistant mutations. There are 3 particular sites. But the part that did not make any sense to us was that, first of all, we knew that resistance arose very quickly. These pesticides were added in 1950, so there's been, say, 50 years at most, but we know the resistance arose before then. Say 50 years, so it's 500 generations. And in that time, not only at resistance at a single site arose, but arose at 3 different sites. And if you look at the population, we saw alleles that have all possible combinations. So one at a time, this by itself, this by itself, this by itself, every pair, and all 3 of them together. And if you think of this hard sweep model where individual mutations arise one at a time, it didn't seem like there was enough time, even if selection was very strong, for the first one to happen, then the second one to happen, and the third one to happen. Just something was not making sense to us. So we decided to probe it a little bit more deeply and looked at the history of these resistant alleles. In particular, bear in mind one possibility that even though we think that adaptation to these pesticides was driven by de novo mutation, it's also possible that these resistant alleles were there present in the population for a long time, maybe because maybe there are some plants or fungi or somebody else who makes some other mimics of acetylcholine, and so flies already have resistance, and so they just used pre-existing standing variation to adapt to human-made pesticides. So we collected flies and looked at this gene in flies from Africa. Just a little bit of background. Drosophila melanogaster did not originate in a lab. It originated in Africa, sub-Saharan, probably in the savannah, and then spread with humans, probably around, most likely with humans around the world. And now they're present everywhere. Wherever there are humans, there is Drosophila melanogaster. But just like humans, this is the ancestral population somewhere Zimbabwe, Zambia, and these migration happened in the last probably 10,000 years. In some cases, what's much more recent, let's say to America, it's probably a few hundred years, probably with the slave trade. So we sampled a number of flies from Africa, from the United States, and from Australia, and we also had these wonderful strains, they're marked here in yellow. They were sampled by people like Morgan and Sturtevant, and they were sampled before pesticides were added to the environment, right? So they're called M strains, and they're old strains, and first of all, they're labeled as old strains, but also they don't have particular kind of transposable element or p element that's spread in the 50s. So if the strain does not have this p element, then you know that it's a very old strain. It was collected before the 50s. So we know the strains were collected before the 50s. And they tell us about this kind of state of the variation after migration out of Africa, but before pesticides. So so I'm going to show you haplotype networks. I hate them, but just it's a very fast representation of the variation. In this haplotype network, so we sequence this thing 1.7 kb region, and we look at particular haplotypes and put them on this network. The individual haplotypes shown here as circles, as nodes. The bigger the circle, the more frequent it is in the sample. The edges represent the relationship between haplotypes. So if they're linked by a single edge, it means they're closer together. And the length of the edge corresponds to how divergent these haplotypes are. The colors again, so the gray ones are Africa, AM are yellow, red, United States, and blue, Australia. So the first thing you see is that there's more variation in African haplotypes, just like in humans, which is probably correspond to the fact that this is ancestral range. And there's a subsampling of diversity at this ACE locus in the United States and in Australia. So one thing I didn't mention, these are all, I'm only showing right now sensitive haplotypes. So I'm only looking at the state of this gene in the absence of resistance, right? So it looks like they came out of Africa, some variants were subsampled, and there are some differences between North America and Australian haplotypes. So now we started seeing what's the origin of the resistant haplotypes in North America, and these are all resistant haplotypes, and they're shown here with numbers. So one means that there's only one resistant mutation at the first site, first and second, first and third, first, second, and third. And you can see that they're all mostly related to this one common haplotype that was present in North America, and it was particularly frequent among these M strains that were there before pesticides were added. So it doesn't look like these resistant haplotypes are sort of ancient versions of this locus that's already been there in Africa and just kind of standing there at low frequency being selected on. Moreover, when we looked at Australia, and I'm sorry, these kind of edges in this network mean that there was some recombination inferred. When we looked at the Australian resistant haplotypes, and this will be easier to see, I'm just going to make the sensitive ones less bright. The resistant haplotypes in Australia were more closely related to the sensitive haplotypes in Australia than they are to North America. So it looks like in Australia, resistance evolves based on what was common in Australia, and in North America, what was common in North America. And it doesn't look like these are ancient variants. So this is a different way to look at the data, maybe more transparent. So, but I'm showing less of the data. So I'm just showing one position of one particular site where resistance can evolve, the first one. So this is the sensitive state, A. The G is the resistant state. And these are the neighboring sites. So these are the common haplotypes in North America and in Australia. And particularly GGC is what distinguishes them. And you can see that the resistant alleles in Australia have this GGC, and in North America don't. You can also see that in North America, you have a single haplotype. You have at least two ones. So even within the continent, looked like it happened more than once based on using different sensitive haplotypes as the background. Right, so the observation is that it looks like it's de novo mutation, arises very fast, you generate a lot of diversity, and you generate these soft sweeps that I talked about before. So the simple theory that Pennings and Hermanson came up with suggests that it could only happen if population size and mutation rates are very high. We wanted to simulate it under wider range of regimes with population substructure, with other complications. And basically what we found that if you take the standard model for Drosophila melanogaster population, say the population size is about 1 million, there's just no way this can happen, this kind of a pattern. You at most get resistance through a single site, and you never see soft sweeps. 99% of them are hard, so 1% of them are soft in simulations. If you bump up population to a larger number, let's say 10 to the 8, so 100 times larger, or 10 to the 9, then it's all very easy. Because the waiting time is shorter, resistance evolves faster, and multiple independent mutations come in population at once. And so it's here, these are independent mutations coming in. The second one happens on the background of the first one before it even reaches fixation, and then the third one arises on top of that. And so if in that kind of situation, you both have, it happens fast, you can generate very complex resistant states, and you expect to see abundance of soft sweeps, and resistance evolves pretty much all the time, just because the waiting time is short. So based on this, we said, okay, well, it might be that Drosophila melanogaster, at least, does not live in this regime, does not live in what we call mutation-limited regime, but in fact lives in a regime where population sizes are large enough that every mutation basically occurs in the population in every single generation. There's no waiting time at all, and so soft sweep should be common. One thing I want to mention is that it doesn't only show up, so we started looking at other examples of adaptation, and in sort of melanogaster, this one particular gene, its resistance, maybe it's something peculiar to artificial selection. Started looking at other examples. So here's one example for an island species of Drosophila. It's a Drosophila santamea, and they evolved. They are close kind of sibling species to Drosophila jacoba, and they evolved loss of the abdominal pigmentation. And so there was a paper where the mutations responsible for this loss were mapped to upstream region of a gene called TAN by Sean Carroll's lab. And when they started looking, they had just two alleles, one in santamea, one in jacoba. And when they started looking at the reason for it, they found there's a deletion in the upstream region. But when they started mapping it to multiple alleles of the gene TAN, they found that there are deletions in all of them, but they're all different deletions. And there are multiple haplotypes. So it looked like there was strong selection for the loss of this abdominal pigmentation, but it did not happen through a single event. It happened through multiple events, right? And again, it seems like a de novo change, again, giving us soft sweeps in multiple haplotypes. So the question then is, why did we think that population size was so small? Like, what's the reason for that? If we think that's completely wrong and we're off not just by a little bit, but maybe by 1,000-fold, which is somewhat embarrassing. The way that people generally estimate effective population size, or population size relative for population genetic processes, vaguely speaking, is based on the amount of neutral polymorphism that you see in the population, which in some sense is a function of the rate of coalescence of different alleles in your population. Basically, how far back in time do you need to wait until you have a common ancestor of these two alleles, if you sample two alleles from the population. So the rate of that coalescence, inversely proportional to the effective population size, or let's just say here just population size, and in some sense can be very sensitive to every single bottleneck that happened in the population fairly back in time, right? So it can, even infrequent but sharp bottlenecks can severely change the rate of coalescence. So imagine if the population size is very large, there's no coalescence while it's large, and then while it's small, there's a lot of coalescence. So the amount of diversity will be determined by basically the time to this time that the population was small in the past. But if you're interested in rapid adaptation now, all the bottlenecks that happened in the past are entirely irrelevant. And you're really only feeling the population size or effective population size that's relevant to this current moment. And so when we study rapid adaptation, in some sense we're sensing what's happening now. When we study neutral polymorphism, we're asking how many bottlenecks have there been in the past, or maybe how many selective events have there been in the vicinity of this particular region we're studying. And so they're telling us about very different time scales. And in general, it's kind of a theorem that this time this population size will always be much smaller than this one. Because, I'm not going to go into details, we can talk about it in future. So, if the current population size, especially when adaptation is happening, tend to be large, then we will be vastly underestimating it by looking at neutral diversity. So we wanted to see if that's indeed the case, then in general, because we're using methods that are sensitive only to finding hard sweeps, we might be missing a lot of adaptive events. And in particular because in a hard sweep, which I'm showing here, so it's single haplotype getting to high frequency, what you expect to see, deeper neutral diversity because you just drag the haplotype to high frequency. You expect to see excess of low frequency SNPs and single frequent haplotype. In a soft sweep, diversity can remain high because you moved multiple haplotypes. SNP frequencies may not be skewed very much, so that signature may be absent. And you should see multiple frequent haplotypes. And so we, again, Penix and Hermison and done some work as well to show that the pattern of LD, in particular, the haplotype homozygosity should be high in both cases. So basically, if you come up with statistics that will be sensitive to the presence of even multiple but frequent haplotypes, then you might be able to detect both these events and these events. And so we came up with the statistics. I'm not going to go into detail right now because of a lack of time, but I can do it after the end of the talk if you're interested. It's a statistic that's a modified homozygosity statistic using whole haplotypes. We ran a scan, and the primary author here is Nandita Garud and Philip Messer. We did a scan in a large panel of fully sequenced Drosophila strains. There are about 150 of them, so a whole genome. And these are isogenic strains, or to a large extent. So when we do the scan using haplotype homozygosity statistic, we find a bunch of peaks. These are 4 different chromosomes in Drosophila. The nice thing is that the known cases of adaptation, so ACE that I talked to you about, shows up as a very sharp peak. There's another known case, viral resistance through Chekhov-1, and another case of DDT resistance, P6G1. They show up, but there are many other peaks show up. I'm not going to show you when we run standard methods like IHS, we find nothing. It's just one big mess. And again, I don't have time to go into it. When we start looking into the individual sweeps, so the 3 known ones are all soft. I showed you one, but the other ones are also soft. When we look at the 50 top peaks, they all appear to be very soft. And this measure of how many adaptive mutations happen for these peaks, how many of them happen per generation, per population, is an order of 1.5. So more than 1 adaptive mutations happen per generation in the population. That's how these sweeps look like. So soft sweeps appear to be truly pervasive in Drosophila, and probably other cases. So this is just very briefly. We did the same thing in malaria. We have 100 completely sequenced genomes of malaria in collaboration with Tim Anderson and Ian Cheeseman from Texas Biomed. And these are scans using another version of haplotype homozygosity, showing you 4 particular chromosomes. And they're chosen because these are all cases where we had positive controls again. These are known cases of drug resistance alleles. And this statistic does give us the right places where we know resistance evolves. If you use statistics like the GMSD, which people like to use, the pattern is much harder to detect. These 2 cases are known hard sweeps in malaria, and there's a bit of a dip, as you expect, but you would not have been able to call it, I think, without knowing where the gene was. Nothing here, which are the 2 cases of known soft sweeps. So I feel like we're now starting to get the right tools to start looking at a broader range of sweeps and finding probably more cases of adaptation starting from de novo mutation, even in cases where they do not leave this canonical signatures of hard sweeps. So, just the conclusion for part 1 is that adaptation in Drosophila, and probably many other organisms, is not limited by availability of de novo mutation. So all organisms where the census size is on the order of 100 million is fair game, even for adaptation by single site. But if you think of adaptation by, let's say, indels or other things, that you can go down to smaller population sizes. So I think soft sweeps will become a rule rather than exception. And we should expect rapid adaptation even by de novo mutation and soft selective sweeps. All right, so how much time do I have? Sorry, just I can adjust my
15
minutes. Okay, so very quickly. So this is adaptation of pesticide resistance, and it's like single gene, single site, or a few sites. We wanted to look at the other extreme and look at adaptation at life history traits, so kind of more broad type of adaptation. So again, flies came out of North Africa, South Africa, 100 to 10,000 years ago, 16,000 years ago, and encountered a whole range of environments, including showing up in temperate areas and probably being completely shocked by discovering that there's such thing as winter. But they're still making it out here, and what's interesting is that there's been this probably already a century of studies of different clines in different phenotypic traits across environmental gradients, and Drosophila, like many other organisms, vary across a lot of different phenotypes, and across, so this is wings, let's say wings are bigger if you go poleward, both in North America and South America. If you look at genetic characters, they also vary. Let's say inversions vary more both in North and South America and in Australia in a consistent direction. So we know that it's likely that there are some adaptations happen to particular areas around the globe, most likely due to the fact that the environments are different. So the difficulty when you study adaptation across space is that it's very hard to disentangle variation due to just demography from variation due to natural selection. And so we wanted to see whether we can detect adaptation occurring on much shorter time scale, and in particular wanted to see whether we can detect adaptation occurring on the scale of a single season. So Drosophila melanogaster goes through about 10 generations over the summer, maybe 1 or 2 generations in the winter. And I'm not going to go into the details of it, but it looks like Drosophila melanogaster can overwinter locally, so they don't remigrate even in areas with severe winter. And there is physiological adaptations to winter, in particular diapause, which is genetically variable. So some strains of Drosophila melanogaster, if you bring them into the lab, you bring them on to cold temperature and short day, they go into reproductive diapause, they resorb the eggs into fat, and they stop aging more or less. They stay in this state for a long time. Interestingly, if you sample flies in orchards, that's work by Paul Schmidt, if you sample flies that early in the spring, most of them can go into diapause when you bring them to the lab. And it's genetically so their progeny also can go into diapause. But if you sample flies later in the season, in September, the proportion of them being able to go into diapause declines, and then it goes up and down. So it cycles. This is in the orchard. If you sample them in urban areas where they probably hang out in people's basements, where the temperature doesn't vary so much, ability to diapause is lower and flatter. So it looks like there's a reasonable expectation that adaptation might be happening even on these short time scales. So we sampled flies across the East Coast. Again, this work, I'll acknowledge people, and in Philadelphia from spring to fall at multiple years. And this work is by a postdoc in my lab, Alan Bergland, and all of it is in collaboration with Paul Schmidt at UPenn. So what we did, from each site, we took about 50 to 200 flies, we pulled them together, and we extracted DNA and sequenced them to about 50 to 200x coverage per site. So this way, we can get on the order of, we get about 1 million SNPs, then we filtered them down to about 600,000 frequent SNPs, with the frequency more than 15% across all populations, and we can estimate their frequencies simply by counting number of reads with 2 alternative alleles, because the coverage is substantial, let's say 100x coverage. So we have 100 counts for any particular allele. so we do this, and first we exclude Pennsylvania, where we're looking at seasonal variation, and we see, do we have any clinal pattern across these populations? We do. So these are, this is the frequency of alleles: Florida, Georgia, South Carolina, North Carolina, and Maine. And we just looking at all sites that systematically go up in frequency, that will polarize them such that they have a lower frequency in Florida than Maine, so they all go in one direction. And this is a measure of population differentiation between populations as a function of degree difference in latitude, and so the probability that the FST between populations is greater than expected if everything was completely panmictic. And as you go, as populations become more and more distant across space, they become more and more differentiated. People have seen this before, nothing new, just showing that this method works. And when you look at the sites that have particular high differentiation, they're present on all chromosomes, 12 throughout 3L throughout X, particular high frequency on 3R, and where there is one particularly big inversion. So again, this is something that's known. All right, so now we're going to do the same thing, but instead of looking through space, we're going to look through time. So again, we have spring, fall in 3 years, and we're going to exclude this particular time point of after the frost. So 6 samples. And again, this is the latitude, so the differentiation across space, isolation by distance. The farther you are, the more different you are genetically. This is differentiation across time. So if you take samples across these 3 years in this orchard through time, they become more and more differentiated. And in fact, after 3 years, the samples are as differentiated as almost half the East Coast. So it's a substantial amount of differentiation that happens over this time. And this suggested to us that maybe we can find SNPs that cycle sort of in the same way the dipause cycles. Let's look for the ones that go up and down in frequency. And we found some, right? So we specifically look for those that go like this. We found on the order of 1,000 cycling sites at fault discovery rate of 35.35. So it's like on the order of 700 sites that probably cycle for real, according to us. And the difference in frequency between spring and fall samples is huge. It's 20%, right? So it's they cycle not by, these are not subtle things. And selection, therefore, at a site is really, the inference is that it's extreme strength of selection at a large number of sites. Very polygenic. Additional modeling suggested to us that at the minimum we need to have 50 sites that cycle in order to explain these patterns, and it cannot be more than 2,000 sites that cycle, because otherwise just selection would not be able to move so many at once. When we start looking to see where they are, so first of all, there are way fewer than sites that are differentiating across space, and they have a very different pattern. They're across the whole genome again. They're not particular regions, but they're not, for example, enriched in inversions the way clinal sites are. They tend to be genic, or in particular, they tend to be coding. So they have higher, there's an enrichment for coding sites when we match these sites with some kind of control set that matched by frequency in North America and frequency in Africa and other things. So these seasonal sites tend to be genic, which is promising. They're marginally overlapping with clinal SNPs in the right direction. So the sites that are more frequent in the summer tend to be more frequent in Florida. But that's very weak. So they seem to be kind of different sites. And we can talk about why that would be the case. So we wanted to, there's a lot of statistics. We started with 600,000 SNPs. We'd end up with 1,000 that cycled. The whole thing could be, we did our best to make sure that it's not nonsense, but who knows. So we wanted to do some additional tests. And one of them was just to see, kind of turning into predictive science, we wanted to see whether the sites we identified will actually be responding to a single environmental perturbation. So we took the 6 samples we called the sites, and then we sampled flies right before and right after the first frost of the year. So this sample was October 15th, then there was a frost November 1st, temperature dipped to 0, then we came back 2 weeks later, there were flies still buzzing around, we sampled them, we did the same sequencing as before and wanted to know whether the sites will go in the right direction. So the sites should go in the direction of winter. They're responding to, let's say, to frost, and frost is one of the things that they're adapting to, or cold temperature. Indeed, they do. Now, this is extremely misleading graph, I admit it, because you expect exactly the same thing if the whole thing is noise and it's just regression to the mean. So in order not to have this to be just an artifact, we compare it with other sites that kind of go up and down, actually, that have this time point as deviated from the average as our seasonal SNPs, and wanted to know whether these sites also go up. And so the control ones, it's like 35% of them go up, and for seasonal ones, 43. So there's significant enrichment of sites going in the right direction as we expected. So this is, to me, it's one of the most direct things that we can do to show that it's not just a statistical artifact. So just a few minutes about where they're coming from and what the whole thing could be about. So we wanted to know whether this is something new. So when we started, we had this maybe naive expectation is that a lot of this adaptation will be adaptation to temperate climates and to winter. And we thought, well, there's no seasonality, or seasonality is muted. Maybe it's dry, wet seasonality in Africa, but it's not, it's nothing like Pennsylvania in January. So maybe it'll be new adaptations. So I wanted to see whether the SNPs that were identified as seasonal SNPs, they're present in Africa. And to our surprise, the vast majority of them are present in Africa. So it looks like, unlike the situation with pesticide resistance, this is an old variation, at least present in Africa. So then it's like, okay, well, is there any evidence that it's old variation in Africa? And for that, we took, for every SNP, we looked at the amount of diversity nearby in the, so we looked at about 70 kb around the SNPs. And we again matched them by frequency with the frequency in North America, frequency in Africa, just so everything's matched. Here we're also matching by genic, non-genic, everything. That's the beauty of genomics, is that you have so many things to just do empirical controls with that you don't need to be very clever about statistics. So the sites that were identified as seasonal tend to have more diversity nearby compared to matched sites, so kind of consistent with what people think of balancing selection. Balancing selection is something that keeps the polymorphs in the population longer than neutral polymorphs that should be there. So it's kind of consistent with long-term balancing selection. And so we say, well, it looks like it might be old and ancestral. And so they say, okay, well, can we go even beyond this? Can they be maintained for even longer than that? Can they be maintained between species? Is it possible that these polymorphs that we see here are also polymorphism in another species, Drosophila simulans, that's separated about 5 million years ago from Melanogaster? And we have polymorphism data in simulans. So we just asked this simple question. Whenever we see a seasonal polymorphism in Melanogaster, let's say AT, do we have also polymorphism AT in simulans? It's the same states, not just the polymorphs, but the same exact states. And again, is the proportion of the time that we see such a polymorphism in simulans greater than we expect by chance? And it is. So again, we match it by everything we can match it with, and there's substantial enrichment of the so the seasonal SNPs are much more likely to be circulating in simulants at the same site compared to matched set. So the next question is that, well, is it possible that they're ancient SNPs, or maybe Melanogaster and simulants now live in very similar places? Maybe they just this parallel adaptation, they evolved the same changes, and so that we see that they're shared more than we expect by chance. But these are not ancient polymorphisms, but de novo polymorphisms in both species. So in order to separate that, we can do a simple test. So sorry, it doesn't show up here. If it's convergent evolution and happen independently in the two species, that the two alleles in the two species should be more similar to each other than between species. Whereas if it's ancient polymorphism, then let's say, again, you have AB polymorphism in Melanogaster, the A allele should be more similar to the A allele in simulants, and the B allele should be more similar to the B allele in simulants. So we take these comparisons and we ask, when we compare, basically, we compare A to A prime, is it more similar than we compare A to B prime? That's our test. And we do it across all seasonal transspecific polymorphisms. And so this is comparison, the distance between Melanogaster and simulants for the same alleles, and this is for different alleles, and you can see that the whole thing is shifted this direction. So the distance for the same is somewhat smaller than between. And I was kind of shocked to see the signature because there's been substantial amount of time that the signature should be eroded pretty much completely, sort of similar to humans and chimps. It's a very similar test to what Molly Przeworski and her group did comparing humans and chimps, and sort of the distance is similar. So at least this suggests that there's very long-term maintenance of these seasonal polymorphisms. So the shocking thing to me is that if it's true that these alleles can change their frequency by 20% in 10 generations in one season, and yet are maintained for millions of years, such that you can see even the same alleles in multiple species, the related species. So, so first of all, how could it be? We think that there's probably, or at least one hypothesis, that it's a case of sort of marginal overdominance, where it's where the two homozygotes flip in their fitness across different regimes, and the heterozygote is never the best, but tends to be kind of in between. But the variance of the fitness of the heterozygote is lower than the variance of the fitness of homozygotes. And if you just multiply all of these fitnesses, you can have a situation where geometric mean fitness, long-term fitness of a heterozygote is better. So there would be kind of a case of heterozygote advantage that shows up across time, across multiple environments. Not sure whether it's true or not, but somehow we need to explain how these alleles can be maintained. But just one more point, that what's clear to me is that whatever the force is maintaining it, it has to be sufficiently generic in terms of ecological terms, because it cannot be about selection in Pennsylvania, right? It's something that's maintaining these alleles in Pennsylvania, in Africa, and in simulants. So the one way we're starting to think of it is that it's possible that during the summer there's selection for alleles that allow very fast growth of the population, so early fecundity and very little investment in somatic maintenance. Whereas in the winter, these alleles are deleterious, and they're alleles that allow for survival when it's dry or cold, there's no food. They are the ones selected. And so it's possible that that kind of a trade-off is sufficiently generic to be persisting across millions of years. Basically, selection trade-off between rapid reproduction and some somatic maintenance. So the conclusion is that we seem to be able to detect rapid, strong, and very polygenic adaptation at seasonal timescales. These cycling alleles are likely balanced for a very long time, and such that they can even predate species splits, and suggests marginal overdominance and persistent life history trade-offs. Just want to thank the people who did the work. So again, pesticide adaptation is all this work of Tali Karasa and Philip Messer. Also talked a little bit about the work of Nadita Garud. Sorry, I don't have her photo. All this work on seasonal adaptation is done primarily. Alan Berglund is really the primary author of all of this work, in collaboration with two graduate students from Paul Schmidt's lab, Emily Bourbon and Catherine O'Brien. And again, all of this work is done together with Paul Schmidt. Thank you very much.
Genes getting duplicated to fix the heterozygotes?
Yeah, so we don't see any evidence of that, and that's one of, so if I can give an introduction to your question, there's an old theory that suggests that long-term overdominance by heterozygote advantage should not persist, because it's always possible to be able to duplicate a gene and create a heterozygote that actually fixes. So we don't see this. I mean, we see them cycling, so we don't need to explain it somehow. Another thing that puzzles me is that why doesn't phenotypic plasticity evolve? Right? They could be just, instead of cycling like this and having balanced selection, they could be just sensing the environment and deciding to turn on the right allele or the right gene at the right time. That also could work. So the feeling I'm starting to get is that the environments in which the pure types are advantageous are sufficiently common. And so the pure types win enough of the time, but they do not win overall. And so somehow that must maintain variation, but I don't know. These are all puzzling questions for me.
It seems like you could test that hypothesis by having multiple samples over multiple seasons in a variety of different geographic locations, depending on how warm or cold the winter was.
So we just sequenced a bunch of samples like that. So we started a consortium of people who started Drosophila, and so they sample flies locally and send the flies to us, and we just sequence them. And so I don't know I cannot tell you what we see there, but it's across the United States and across Europe as well. So we'll find out. But that's a great question. Whether it's not clear what we expect, to be honest, because different alleles could be cycling in different populations. So
Right. If they're the same alleles, though, then you'd only expect that heterozygote advantage from that particular allele should only work in that particular location, right?
no.
If it's
I don't-
related to temperature.
No, we don't know what it's related to. I think that it does not relate to temperature. I think it relates to something much more generic,
Okay.
like ability to not die without food. Right, which is common both during the dry season in Africa. I just came from Zimbabwe. It's not a particularly nice place to be in the dry season. And so in temperate areas, it's dry, cold, and hungry. And in Africa, it was dry, hot, and hungry. So
Especially since it's diapause, so it
Yeah.
makes sense because it's got a lot. I mean, diapause is a response to the scarcity of food. So
That's right.
Probably many of the SNPs are in metabolic genes, and that's helpful in cold temperature. It's helpful for any
that's right.
adverse conditions.
That's right. So that's what we're thinking.
If the probability of soft sweeps is related to the size of the population, as you indicated, shouldn't this mean that other groups like vertebrates would have a much lower probability of soft sweeps? Because they have much,
It should, depending on like in mice, I don't know what
well,
we
I mean,
expect.
in general, the population sizes would be different order of magnitude.
Yeah, but I don't know whether it would be below what. So in Drosophila, right? So step back. And in Drosophila, we expect soft sweeps whenever populations are large, even at single sites. In vertebrates, so if you have adaptation through gene loss, then the total number of mutations that can kill a gene multiplied by whatever population size is, let's say 10 to the 5th, could still be greater than 1. So in principle, yes, but I don't think anybody has done a systematic study.
Yeah. I'm sorry, was there a question? Go ahead.
It looked from the plot like your seasonal SNPs might be enriched near telomeres. Is that the case? Why do you think that might be?
We did not find that mechanism. So it's a little bit, and I think it might be related to recombination, if there is. But when we looked at it, there's slight enrichment in regions of low recombination, as you would expect.
It's very striking that you're measuring seasonality in Pennsylvania, but arguing that this sort of selection has been going on over 5 million years across very, very different environments. So if that's the case, and it's tied to something like being able to do well when not much food is available, you might be able to recreate that by applying selection in population cages. Have you
Paul Schmidt just started the experiment, which is entirely insane. It's just the whole basement became one population cage, and we're taking our flies through exponential growth with unlimited food for something like 5 generations, and then we're just going to starve them to death and sequence fractions. and we'll find out. I'll tell you in a few months.
You do that with
Smell.
That was 6 dimensions?
Oh, 6 dimensions. We're studying through time, through space, in 2 species.
Okay.
And it was funded.
Great. Well, thank you very much, Dmitri.